Thank you very much for the kind invitation, and for letting me talk actually about a project we started a little over a year ago, reviewing prediction models for diagnostic and prognostic purposes related to COVID-19. My name is Maarten van Smeden. I'm a statistician by training and an assistant professor in epidemiologic methods in Utrecht. In the background, you see the hospital I used to work. I've been working from home for a long time now. We can go back slowly, which is good. All right. So obviously, we're in a world that is becoming more and more digitalized. I'm sure all of you notice this. And if you ask a patient how he looks at modern medicine, probably they think about all types of fancy equipment and machinery, and maybe they even say artificial intelligence. And I'm sure most of you who are statisticians will recognize this, is that there's a lot of algorithms going on in our field, and a lot of enthusiasm. And at some point, people often realize that it's often much more complicated than they think. I think this is just a small grab from the medical literature. There's a lot of machine learning these days going on, from multi-center machine learning studies to, especially the word leveraging is used a lot, leveraging machine learning techniques, deep learning, roadmaps, all kinds of stuff, all related to machine learning. And then COVID came, and it was not very different than, I guess, the rest of the medical literature. We saw a lot of machine learning, artificial intelligence, right from the start of COVID. So in March, there were newsletters that said that people deployed systems with doing over 1,300 screenings per day. In March, at that point, we almost had no data yet. AI was battling COVID in Italy. I guess you all remember that at that point, really at the start in March, the problem was really in Italy. And also in the UK, you've seen a lot of actually also good stuff coming from the UK. And this is not only just in theory. These algorithms that have been built around COVID been used to, for example, make shielding lists, for good or for worse. And there were also many algorithms actually developed that got the sign of approval from the FDA. So just to give a sort of a background, maybe most of you know this already, but why are we building those diagnostic and prognostic models? Well, the idea is that you can use those models to support clinical decision-making in individual patients. And well, just in general terms, it's simply, of course, combining and giving appropriate weights to input, which can be signs and symptoms from patients. It can be lab results. It can be demographics, it can be CT images, it can be many, many other things. What we typically want from those models is to get a risk, a probability of the outcome, the diagnostic outcome or the prognostic outcome for an individual patient. And it doesn't have to be complex at all. So this is very simple decision tree, based on just three variables, LDH, CRP, and lymphocyte counts, which can be used to get an estimate of the risk of death. This is another one that was quickly developed, COVID-19 early warning score, based on signs of pneumonia on the CT, history of contact, fever, age, sex, Tmax, NLR, things that are relatively simple to measure. And then you get a sum score. And in this case, some score higher than 10 would indicate a highly suspect patient. And there are more advanced methods like the 4C mortality and 4C deterioration, which are based on, actually, these are not too complicated. These are based on regression and some spline functions. And if you fill this in for an individual patient, you get the risk for mortality within that particular patient and or the deterioration, the chance that this patient will go to the ICU or indeed die. So these are simple models, but there are also more advanced models where they use chest CT with typically deep learning algorithms to do some segmentation and in the end, some sort of quantification of the risks. And if done right, these prediction models, the idea is that they can help improve care, increased diagnostic and prognostic speed. They can help with some sort of uniformity, especially at the start of the pandemic. Of course, medical doctors had to use their own judgment. Sometimes it's better to use models to get some uniformity. And the idea is, of course, that also these models can help with the accuracy of the predictions at reduced costs. And the idea of these models, especially in COVID times, was that these models can help allocate and prioritize these scarce health care resources that we have. But we also know from earlier studies that very well-known prediction models like SOFA and Apache 2, which are used in ICU typically, were actually performing very badly in patients with H1N1, so another infectious disease. We probably all know this kind of examples where algorithms can easily be fooled. And this, I like this quote, claiming that the classifier trained on zillions of human-labeled images containing cats and no cats is recognizing cats is just stupid. A human can see a handful of cats, including cartoons of pink panthers and lions and tigers and panthers, and they can not only recognize many other types of cats, but even if they lose their sight, might have a pretty good go at telling whether they are holding their moggy or their doggy. So there are many of these examples. Of course, I like this one, adversarial example, where on the left, an algorithm classifies this as a cat with 88% probability. And now the question to one of you, anyone who dares, what do you think the other picture on the right is classified as? Any suggestions?
An iPad?
An iPad, all right. It's close. Well, maybe. It's closer than to an iPad than to a cat. Yeah. Any other?
A rabbit?
All right. Well, it was guacamole at 99% probability. These are, of course, funny examples, but there are more realistic examples. I'm not sure whether you know this one, but this one was a paper published in Nature, one of the first ones where they showed that deep learning was actually very capable of classification of skin cancer with deep neural networks. And they got a lot of citation, a lot of attention, but at some point it became clear that actually what the algorithm had learned was recognize a ruler in the images to diagnose it as skin cancer, malignant skin cancer or not, because the pictures, of course, with the malignant tumors had a ruler in it. And this problem was also identified, but of course got a lot less attention than the earlier paper. This is all very nice in theory, of course, but coming back to actual medicine, I like this paper a lot, where they did actually a trial, a very, very nicely designed one, where they looked at the impact of a deep learning assistant on cancer diagnosis. So what they did was actually let pathologists decide on whether something or histopathology was cancerous or not, but assisted by an AI algorithm that said yes or no. And they simply switched the label sometimes. And what you can see is that the odds ratio of making a correct diagnosis, if the algorithm gives the correct diagnosis, actually improves sort of the chances of getting a correct diagnosis in odds ratio of 4.2. But if it's incorrect, the model diagnosis becomes much, much less plausible. The problem, of course, is here that prediction models, if you apply them and you let them, even if they don't make the decision and you leave it to the medical doctor, giving them information that is wrong predictions, can actually make the medical doctor perform worse. Now, of course, this is all risk-benefit again, right? A good model might have benefit, but a bad model might actually do more harm than good. All right. So getting to this research that we have done, we have published this paper actually at the start of the whole pandemic, British Medical Journal, with a pretty small team at the time. Just to give you an impression, what we did, we had this idea of that we had to do that based on these news, based on the news that a lot of these models were actually implemented in practice. We decided in March 13 to do this systematic review with the people I just listed. We screened 1,900 titles, included 15 studies describing 19 models, and we submitted the paper in the end almost just over a week later. A lot of evening work by the team, and then we did an update actually 2 days later, because of course the amount of publications was so rapid, at least still, but also at the start of the pandemic. Received a peer review at the 28th, replied to it on the 30th, and got it accepted at the 31st. So that was a very rapid, at least for my standards, very rapid review process with actual review comments, not accepted out of kindness or something. So 18 days was really a sprint, and we're actually quite surprised that the BMJ invited us to do a living review, which was their first living review ever, which made the sprint into a marathon. Because we're now at update 3 that was published in February, and we're now finalizing update 4. And I will present the results from update 3, because we don't have the update 4 results yet. But one thing you can see probably is that the team became a lot larger over time, because we needed a lot of hands, because it's a lot of models. So why did we do this? Well, the aim was just to understand what was available in terms of prognostic and diagnostic models. So to have an up-to-date overview of COVID-related prediction models. That was the aim of our living review, and to give a critical appraisal of each of those models. The general question was, which models can we sort of support or identify as perhaps useful in clinical practice? We didn't want to be the statistical police. We really wanted to help medical doctors decide which model to use. So fortunately, we were able to do the systematic review quite quickly, because there were quite good tools that we could use. And one of them was the CHARMS checklist, which sort of simplifies and sort of simplifies which data to collect from this kind of systematic reviews for prediction modeling studies. We also use the so-called ProBust tool, which we use to assess the risk of bias for each prediction model. And you do that risk of bias assessment in 4 domains. First of all, the participants domain, so whether it is a good representative sample of hopefully consecutive patients, which is very important if you do risk prediction modelings. Whether the predictors are measured at the same time, that kind of questions. Outcome, whether it's assessed independent of predictors, also at the same time. Whether there's a clear time horizon. And an analysis domain, which is about sample size and considering non-linearities and missing data, that kind of stuff. Any questions about this so far? All right, so we found prediction models related to prognosis and diagnosing sort of 3 main types of models. So the first one, especially at the start of the pandemic, we found a lot of models that were interested in using mostly deep learning algorithms actually to diagnose patients. This was also at a time that, of course, tests were not widely available yet. And there were also a lot of models that tried to classify COVID-19 pneumonia. And what we have seen more and more over time is a lot of models that try to estimate the risk of people dying from COVID-19 when they were submitted or need respiratory support or in some way deteriorated over time when admitted to a hospital. What we've seen much less was models that try to quantify the risks of healthy persons getting infected or getting at some point severe COVID-19. We'll come back to that later, why that is. So in the last review, we used 43 reviewers. We all did duplicate reviews, independent reviews, and then tried to make a judgment from that. Again, extraction based on the charms and probist checklist, and each prediction model. Some papers present more than one prediction model, and we assessed each prediction model here separately if there was more than one developed or validated. So this is sort of the overall schematic. We started off with 37,000 records that we had to screen of work. We included 170 studies with 236 models. 11 of them models to identify these subjects in the general population, 180 diagnostic models, and 107 prognostic models. I have to say that the last update that we did, so this update is actually looking at the literature at July 2020. So already in July 2020, we had 236 models. You can imagine our new review will more than double that. So the results, most of them were single country data. And at that time, most of the models were actually from China. Also quite some models that we didn't know the origin from at all. Sample size was the median a little bit over 300. The number of events was on median 70. And I don't have to explain you that that is quite low. But also a lot of variation, of course. Most of them were for patients in hospitals. Why? Well, very simple, because this is where we can get the data the easiest, I think. Some patients at triage centers, fever clinics, especially in Asia, where they have these very special fever clinics that we don't have yet, I think. Patients in general practice, much less, and also a very large group of patients that after reading the whole manuscript, you have no idea where they were coming from. Many of them for confirmed or suspected COVID-19 patients, and also again not negligible group of patients that we had no idea for where they what the target population was. And I think another interesting thing, approximately half of the papers actually provided a web calculator or regression coefficients that you could actually use. So you could also turn it around, of course, is that 50% didn't present anything. So you could reproduce or actually use the model. So I'm not a big fan of pie charts, but in this case, it looks pretty nice because it's nicely shows nicely that it's not only logistic regression models that we looked at. There was also a lot of neural networks and deep learning, especially in the diagnostic papers. And also Cook's proportional hazards models, support vector machines, random forest, that kind of stuff. So they make a very nice equal pie. So to give you a sort of an idea of what kind of predictors did they actually use to make those predictions for prognosis, it was of course age and sex, but also comorbidities, temperature, heart rate, respiratory rate, oxygen saturation, blood pressure, images, features, lymphocyte counts, and C-reactive protein. These are the most commonly used predictors that we saw. Of course, a lot of variation in that. Diagnosis, flu-like signs and symptoms, cough, sputum, contact with other COVID-19 cases, and a whole and some other variables that I won't mention anymore. And to give you an idea of what kind of areas under the curve we found, of course, we found the whole range. And what is interesting, I guess, to see is that we found a lot of models with areas under the curve around 0.99 or close to that, which is obviously if you're trying to predict stuff and you come in the medical situation, you come around areas on the curve of 0.99, there's something wrong because it's prediction is not that easy in medicine. So what we have seen also is that quite a lot of papers was actually impressive is that a lot of models actually got internal validation, which compared to earlier prediction modeling reviews, you often see that studies don't do any type of internal or external validation at all. Most of these papers did that, which I guess is a positive thing to take from this. So this is sort of the main result of our not so positive result from our review, which is the risk of bias assessment. In the left upper corner, you can see sort of the overall judgment, which is all of these models have a high risk of bias, except for two, which have a unclear risk of bias. And you can see that that's more or less the same across the types of models that we included. And reasons for that was, for example, in the participants domain, well, what we often found was that we had no idea about the study design, which type of patients were actually included. Predictors were actually quite difficult to score, of course, in imaging, where you talk about not about predictors but about features, which are sort of well developed out of an image. So we scored at a lot of unknowns in imaging studies. Outcomes were sometimes a problem. Subjective or proxy outcomes were often used. And I think the biggest issue, also if you look back here, is in the analysis domain. This is where almost everything turns red, which has to do with very small sample sizes, overfitting and optimism that isn't corrected, or inappropriate or incomplete evaluation of performance. So one of the things that was rarely assessed was the so-called calibration of these prediction models. This is a calibration curve. I'm sure most of you will know this kind of stuff, where you just depict the observed risks versus the expected risks, and you hope that the lowest curve is very close to the reference line, and in this case it's slightly deviating. This kind of information is very important if you're interested in quantifying risk for individual patients, because they actually reflect how adequate the risk estimates are. It's not something you get directly from an area under the curve. So the conclusion, quite dark conclusion of this third living review, is that models are all at high risk or unclear risk of bias, and we do not recommend any of the current prediction models to be used in practice, but one diagnostic and one prognostic model originates from higher quality studies and should be independently validated in other datasets. All right, so this was our conclusions, and sort of backed up by other studies as well. This is a tweet by Eric Topol, generally a big fan of AI. It was an interesting situation where there was one prediction model developed, and 3 other teams tried to replicate it, and all failed to replicate that particular prediction model. Was also involved in a actual systematic evaluation, so an external validation of 22 of the prognostic models in quite a large sample from the UK, dataset from the UK. And our results were quite strong. We found that so single variable oxygen saturation or patient age are indeed strong predictors of deterioration and mortality. And none of the 22 models that all included many variables were actually better than just one of those variables. And I will show this in the next slide. These are net benefit curves. So the delta net benefit compared to these models, and so mortality models at the bottom and deterioration models at the top, you find that they're rarely better, these models, than these single predictors alone, which is a very alarming finding, which sort of supports our systematic review that most of these models are developed very poorly. And we're not alone. This was a recent systematic review who sort of did the same thing as we did, but then looking at 415 deep learning algorithms. They found also that none of the models identified are of potential clinical use due to methodological flaws and underlying biases. So sort of the same result. And this has had some impact if you look at what the World Health Organization is saying at the moment. What they say for clinical decision making is that for patients with COVID-19, severity assessed in a clinic or hospital suggests clinical judgment at the moment, because to guide patient management decisions rather than the available prediction models for prognosis. Of course, this is quite a big failure. It makes me very sad. Anyway, our review, of course, has limitations. Other models are available. So we've heard, of course, we don't know how often that's actually done. A lot of models that were in-house developed, proprietary algorithms, that of course don't get any scientific reports, are just implemented in practice, which are obviously outside the scope of our review because we cannot get information on that. But the little indications that we have, like this paper, is that those models are not doing much better. So now the positive thing: how can things improve? Well, one of the problems was the very small data sets that were often used. And it's not like there's not guidance on how large the sample size must be. These are a couple of papers, recent papers, that give very concrete guidance on how to calculate the minimal sample size required for prediction models, both development and validation. Another thing is, of course, the calibration that is often ignored. All those models are typically used on an individual patient to give an estimate of their risk, and without looking at the calibration, you're in trouble. And also using risk thresholds, especially when it comes to making medical decisions, have to be handled with care. This is stuff we have explained in 2019 quite elaborately. But it's not only this. Another big issue, I guess, with these prediction models is the lack of representative data. So at least at the start, it was very hard to get large data sets of COVID patients. Another thing was that if you look at those papers, often it's not very multidisciplinary. Often it's medical doctors doing it, or a group of statisticians. And I think it only works developing such models if you do it multidisciplinary. And also sharing of models. So 50% of those models that were actually developed couldn't be used at all because there was no code or no regression coefficients available to actually apply the model. Another thing is that what I guess a lot of people don't realize is that before you have a prediction model in practice, there's a whole pipeline of possible failures. The wrong target population, expensive predictors. Of course, if you have expensive predictors, nobody's going to use your prediction model. Discrepancies between development and use, no transparency about how the model actually comes to predictions, no validation, poor development, insufficient reporting. Of course, there's not really an incentive to do a validation study if you have data. The way you become famous is develop your own model. And usually small studies, especially those validation studies. And what is often not realized is that there's actually regulation applying to prediction models. Medical device regulation often applies to prediction models. You have to think about software and hardware, model updating and quality control. And that's before we start talking about how we can make prediction models actually user-friendly. You often see that prediction models are just too time-consuming or developed on prediction outcomes for which no prediction is actually needed. And the low-hanging fruit here really, at least that's my interpretation from the review, is the reporting should be better. It's incredibly poor very often. And there is, for prediction modeling studies, there's the tripod statement that's very easy to use. And not only for prediction models, of course, most of you will know probably the equator network with all types of reporting guidelines. I think as statisticians, we really have to push the applied people to start using this, because it's such low-hanging fruit. We're not there yet. We're working on the fourth living review. We have been looking at 90,000 new COVID articles, and we will be preprinted our article on our website. And we have also been doing an IPDMA with external validations of these COVID prediction models, which will soon submit, and you can already imagine the results. Most of these models, regardless of the data that we have, so across actually the world, we have data sets from Asia, from America, from Europe. Almost all these prediction models that can be used are actually doing pretty poorly. We're not sure yet why that is. We've been also working on a WHO commissioned project on the validation and update of these models for lower and middle income countries, because this is where COVID-19 will definitely be a problem for the years to come. We've been also looking at a project comparing preprints and peer-reviewed prediction models, sort of a side project that we're interested in and whether there's a big difference. But I don't want to only share the negative news. There are some positive examples. There's the Q-COVID model, which was actually recently redeveloped, updated. But the Q-COVID model and I think the ICERIC foresee mortality score are two models that I think stand out in an otherwise a pretty poor field. So we've been doing this IPDMA. So if you're interested and you want to know more, we have a website. And if you really want to have more details per model, we have opened our data from our living review. So you can look at every model and how we scored it and disagree with us. I welcome all comments on that. And I also want to plug this website by our colleagues from Kiel, Richard Riley's website with excellent information about developing prediction models, reporting, controversies, IPD, and that kind of stuff. Excellent website that you should check out if you're interested in this. And I also want to highlight it's I think very important is this is really a team effort. We've had a lot of reviewers working long hours to make this possible. So I'm very glad that they all got involved in this. Last thing I want to say is my slides are already online. If you want to look at them on SlideShare. And special thanks to Lauwe, who initiated this review with me and made some of the slides, and Georg Heinze, Ewout Schuit, Gary Collins, Maarten de Vos for some of the materials that I shared today. Thank you very much for the attention. Happy to take any question.
Thank you very much, Maarten. I could just ask everyone to thank Maarten in the usual online way. Or even better, with a real hand clap. So that was a very interesting, if slightly alarming talk. Has anyone got any questions to start off?
Sorry, I'm trying to work out how to remind myself how to put a hand up.
No, that's fine.
Yes, very interesting talk. I must confess I'm not very surprised. And so it's really a couple of observations. One, yes, I wholeheartedly agree with the problem of lack of multidisciplinarity. And the second is, are there any guidelines for reporting of epidemic prediction models? Because the things I've been asked to review are seriously alarming. You know, people predicting what will happen, including Africa, on the basis of one private clinic in the Congo. And that represents the whole of Africa. So perhaps Maarten can reassure me there's already a wonderful guideline.
I'm not sure. So these models are really outside of our. So our review was really about the individual, but I've also seen those models. Of course, we've all seen those models, and that's really not my expertise. And I share your concerns with these models too, but I've not seen any. But maybe you can contact the Equator Network or Gary Collins in particular. You will know. If it exists, he probably knows.
Any other questions? I couldn't see any hands. Apologies if I've missed them trying to scroll through. Feel free to speak up if you do have a question. Oh, Heather.
Hi there. Thanks for a nice talk. Obviously, at the beginning of the pandemic, there will have been a lot of very small studies for obvious reasons, but now it's been going on for so long, you'd think it should be possible to get much larger studies and therefore hopefully get more accurate prediction. Is that happening? I mean, those ones that you recommended as being possibly the best, are they quite large?
Yeah, these so-called good models are indeed from large initiatives, all UK, by the way, because this is where it's best organized, probably. So these models from the ICERIC database, I think it's 70,000 plus, and the other one is the Q-COVID, which is a couple of million people from the GP registries. So, but these are really the exceptions. And my own experience, it's impossible to combine data sets. So our own IPD, it has to be two-stage because people just don't let go of their data. So this is something that really stood out to me, is that even in a crisis like this, it's absolutely impossible to get data, to get high quality data from multiple centers in one place to do the analysis. So you're doing imputations on different machines, and it becomes much more messy. So what people do is typically develop their own model on a very tiny data set, and all problems related to that.
Question from Daniel.
Hi, thanks very much. I noticed that the high quality models, that developed some primary care data, and I just wonder if you could talk a bit more about data fidelity in primary care. You're relying on GPs as you're not only helping patients, but also collecting your data, and if you had any thoughts about how that works and how successful it is.
Yeah. So I think so we scored this low risk of bias, and my own experience with GP data is that it's quite messy and very selectively reported. And I think one of the concerns with the that I still have with these Q-COVID models is that, of course, it's based on patients presenting at the GP, and it's sort of extrapolated to the rest of the population. But to be honest, this is the best we have at the moment. But this is always a big problem. I think they did a quite a good job with the data they have, but there's at least in my view, a serious limitation. But feel free to comment on that.
So, in the absence of any other question from anyone else, I had a quick question. So this is a living review. How long do you anticipate it will live for? I mean, the rate of articles seems like it's still increasing, right? From what you said. I don't know if you have any thoughts on kind of where the future direction of travel is.
Yeah, so this is recorded, right? But
we can stop the recording.
No, it's fine. But I'm thinking that this might be the last of our updates, because it's a massive amount of work, and it's a massive amount of bad studies. But we are working on updates and development of models for the lower and middle income countries. That will be an interesting direction, I think, because they don't have the vaccination that we currently have, so they would very much benefit, I think, from these models. We're also looking into sort of prediction models in the elderly, but these are all sub projects. But I think the main project with this. I think this will be our last update, because it's just too much work. We will be at I think at 500 models when we publish this last update. And to be honest, I cannot motivate our reviewers anymore to look at this stuff. It's really hard for them, really demotivating.
Oh, understood. Thank you. So I gather Chris and Sandra's iPads, I don't know if this is Chris or Sandra or both on either, also may have had a question. I think you may be on mute though. Right.
I think, right. I just wonder if we're always including the right variables in our models. Did that come across?
Yeah. So of course the variable selection is a very interesting question. I think the question, of course, is what is right, especially in a prediction point of view. You're sort of in the situation where age and sex does a lot of the work for making a prediction, and everything you get on top of that. That's also what came out of that validation. Sort of subject to the law of diminishing returns. It's a nice paper by that makes that point by David Hand, famous statistician. So I'm not sure what are the right predictors, but what we have at least seen is that a lot of models developed are using the wrong predictors in predictors that are unavailable in medical practice. So of course you can make very fancy biomarkers, but if you cannot measure those biomarkers in practice, you're in trouble because your model becomes non-applicable. I'm not sure whether that answers your question.
Thank you.
And I think Daniel had a further question.
Hi, sorry, being greedy. I'm very sorry. I must admit I haven't read through the Living Review, so you may touch on this. One of the things that in the UK we seem to focus on was using frailty as a triaging tool. Almost, is that something that you pick up on in your review? And if so, or if not, do you have any thoughts about sort of widely already used measures being repurposed?
Yeah, so the question about frailty is very interesting because of our project related to the elderly. This is exactly what we want to look at. We just started that project, by the way, last Friday. Just got money for it. But I haven't seen many prediction models, to be honest, that used any measure of frailty, especially in the elderly. I'm sort of concerning in some sense, and is that there was really limited attention to the elderly, although we knew that this is where the biggest problems were. So I don't have that result yet about frailty. We have to look into that more in detail, but was not among the most commonly used predictors.
Thank you. That's really interesting. Thanks.
See that Jane has both found and decided to deploy her raised hand. So Jane.
Sorry, thanks. Quick question. I'm amused that say age and sex are the most important. We haven't exactly needed the genetic revolution for that. And yet, as far as I know, they're not even routinely used most of the time in things, for example, the dose of drugs, apart from child versus adult, those things are just ignored. Sorry, that's more of a comment than anything else.
Yeah, I agree. Yeah, it's interesting that some of these models don't use age and sex, and then the performance goes way down. I'm not sure why people do that, but these are, well, that's maybe a discussion for later time. But for medical prediction, age and sex often do the most of the work. If you look at R squares and stuff like that.
And a question from Chris.
Yeah, sorry, I've also found my hand. So this all focuses on kind of direct clinical, prediction, right? So risk of COVID or risk of deterioration. But I was wondering if you'd done any thinking or work on the more operational side of COVID modeling, because similarly large numbers of models being developed to model things like where you should deploy ventilators to, or how to allocate resources, and those have clinical input, impacts on staff and on patients, maybe even over and above individual models operating in small areas on particular diagnostic pathways.
Yeah. So I think one of the motivations to do this is because we think these models are used for exactly that. So when at the point that our third update or something, there was this scare that there were not enough ventilators at some point in some countries, at least in the Netherlands, and I think also in the UK, is that these models would be used to sort of prioritize patients in sort of the reverse, right? So that the chances of dying, if you have a high chance of dying, maybe you shouldn't get the ventilator. That was the sort of the motivation also to continue this. And yeah, one of the downsides of our review is we have no clue to what extent these models actually have been used. That's always a question that is in my mind, and I think in most of the reviewers' minds, is we've said that all those prediction models are not great, but we have no clue about how much they have been used, and we have some information from newspapers that they have been used, and people claim they have been used, but we have no actual numbers. And I'm not sure whether they have been used for triage decisions, whether people are sent home because of those models. Probably they are, but I'm not sure.
And there's a big difference between saying you use them and actually using them, right?
Yeah, probably. But this is one of those areas that sort of needs research, right? It's very, very interesting that a lot of models are just implemented in practice without any publications, and we have only looked at those models that are actually published. But we don't know. It looks like the Wild West to me, but maybe it's all not that bad.
We have time for one more question if anyone has one. No, well, if not, then all remains is to thank Maarten once more for a very interesting talk. Thanks for coming online from the Netherlands. We appreciate it greatly. And thanks again for a very nice talk.